All right, so first off, I already mentioned to you this idea that if you do not already have a project that you are in love with and you want to try to find something that's maybe a little bit more interesting to continue doing background research on, then I'm recommending you go to the high school level Science Buddies projects and read through them. I'm not going to promise that you will absolutely get a yes from me for it. I will say that the vast majority that I've looked at, I'd say 85, maybe even 90% of them, I would say yes for. Some of them might require additional like IRB paperwork if you're going to try to use dangerous chemicals and you want to actually participate in science fair. But for the most part, you should be okay with most of them. Remember that for the ISP, you have to demonstrate mastery of variables. So you have to be able to tell me what your independent variable is and your dependent variable's response should be. You have to have a hypothesis. So I'll give you an example of a project that I thought was really cool and you would learn a lot, but is not something that would really fit that description necessary for variable manipulation. It was on genetics. It involved if you had already done some kind of DNA testing, then you would be able to learn about haplotyping and haplogroups and apply that to your own analysis that you received. While that is very informative and really interesting, what's your IV? What's your DV? It makes it in such a way that while it's very interesting and you would learn a lot, it's not really fitting the criteria for testing that I need to have for this particular project. So when you're looking through it, make sure that you can identify what the independent and dependent variables would be. In those cases, then it would probably get an okay approval from me. So what are you going to do if you don't have a project you're in love with? You're going to?
Science Buddies, high school level.
Thank you. Okay, good job. I like the answer to that. All right, now a couple of things that people have been asking me about, just wanted to make sure is totally clear. Your whole project is going to be due the first week back from break, okay? Now, that means you can exit. Once you have approval from me for your project, you are allowed to execute it whenever you want. And we've all agreed that not all science fair projects or ISPs are going to require weeks and weeks and months of preparation, right? There are many that are going to be executable in a single evening or weekend. We all know this. This is not some kind of news flash for anybody. So let's not get into some kind of argument with, oh no, science projects, they take so long. They do not always take long to execute. You know what does generally take a long time to execute though?
Background research.
Background research. That does take time. That is the lengthiest portion of any research project that you're going to do. The actual execution of the experiment, once you have all your materials together, it's generally not that time consuming. Now, you still have to get all your data together and you gotta put it all together that way, but as far as the actual data generation, we accomplish that a lot of times in an hour in this class. So I wouldn't expect it to be that different really for your own individual project that you would do. If it took an hour or 2 hours to accomplish something like that, that would not be surprising to me. But the background research, that requires more time. So let's talk about that too. Your background research paper is going to be due Friday, December 13th. If you'll notice, your unit exam is due Tuesday, December 10th. So that actually gives you several days after your last exam and before final exams. So final exams are that following week. So if you were to look at a calendar like this, okay, so if we were to look at a calendar like this, here's your last unit exam, this is December. Here's when your paper will be due. And here is when your final exam will be. You guys are third period, so you would be on the 18th. 2, 4, and 6 would be on the 19th. So you've got time still for the background research paper, but you've also got time to even complete the project. If you would like to complete your project and you have approval to do so, get it knocked out, have it done so it's not hanging over your head over winter break. Okay, so any questions about due dates? All right, now when I'm talking about the whole project being due the first week back from winter break, there's two aspects that you're going to have to demonstrate. Well, really three. The first aspect is going to be a formal lab report, just like you've been writing all semester long, same kind of formal lab report. That formal lab report is just going to be, the only difference is the background research paper that you're writing that's going to be due here. That background research paper gets inserted into your formal lab report, so you don't have to have any additional work for your background research. So instead of just a little paragraph, you're going to have your 2 to 3 typed pages of background research that you'll have in that formal lab report, so it will be longer as a result. The other difference is you are going to have pictures. I am going to require pictures, and we do need to discuss what that means exactly, because when I say pictures, I mean pictures of you with your materials. These are all my materials. Oh, pictures of you doing, this is me doing my experiment. Take a picture with your face.
Aww.
Yes, I have to have faces. Do I care if there are other people in the background? I don't care who's in the background. It can be groups of people. That's totally fine. But I want to see pictures of you with your materials. I want to see pictures of you doing your experiment. And this is going to be a grade-changing aspect because I'm going to make sure that the picture portion is worth at least 10% of your grade, which means you can't get an A unless you include those pictures. Do you want to know why? I want those pictures. So many reasons. Just a second, you have a question?
How would I do it if I'm just doing like data measuring?
You with your computer.
Oh, okay.
Yeah, this is me and my computer, and then have a nice little screenshot, like where I'm actually able to see that you're doing that and not just sitting at a computer. Yeah, so many reasons why. Number one, I don't want you basically turning in data from some project you did in previous years. So, you know, you did a really cool project last year and you want to try and, you know, pass it off as this year. Well, you don't look exactly the same as you did last year. Or the year before, or the year before that. And you certainly don't look like total strangers who may have done this project and you found online somewhere. So, yes, pictures are going to be very, very important. And so I am aware of that, I am requiring that. That's something that you would be attaching with that formal lab report that I'd be able to, you know, actually see your face. Yes, it has to have a face. Okay, so that formal lab report, that's due basically when you get back from break, with pictures. Speaking of pictures, let's talk for just a second about the journal that you need to be taking notes in along the way. So your journal needs to also contain those pictures. So the same pictures that you would print out with that formal lab report, you can actually include in your journal. Your journal can be digital if you would like it to be digital, or it can be on paper. It doesn't matter to me. In order to really submit it, probably the best way, and also make sure that it's with your project if you are doing science fair. But since I need to see it from everybody, it needs to be in at least a small 3-ring binder or a 3-prong folder is totally fine. So either one of those things will work. So you can put paper in there, or if you're doing a digital version, that's fine. Just print those pages out and you can just secure it in your little 3-prong folder. So what do you put on there? So the pictures that you take, yes, that's one aspect of it. But even starting now with the background research that you're doing, the background research that you're doing requires journal entries as well. What does that mean? Well, what websites did you search? And what did you do with the information that you found at those particular websites? So let's say you're doing some background information one day and you're going, oh, look, I found this really cool website. It has some really good information on it. I'm going to print this article out. How does that look in a journal entry? You're going to have a date. You're going to write, did background research for a project, found this website. You write the name of the website down. Printed off this article. You write the name of the article down. If you don't print out the article, then you can say copied and pasted, you know, the diagram from here into a Word document. Something along those lines where you're telling me what you did with that information, okay? With a date and a time, so that that is an actual record of what you did. So additional to that then, every single step that you take in accomplishing that project also needs to have a date and a record of what you did, okay? This journal is one aspect then of what you'll be turning in. I should expect this journal and this formal lab report when you get back from break. Both of those things. Any question about both of those requirements? Yes?
For the journal, do you have to like put like when you made like the lab report? Like when you did it?
Why not? Certainly not gonna hurt, right? One thing that never hurts with journals is including more information than is required, right? So whenever you in doubt, include it. It can't be wrong to include stuff. I'm sorry, the only wrong thing to include would be like other assignments for other classes, or hey, just did homework for English class. Okay, don't put that kind of stuff in there. But anything related to the project, totally good. Okay. Guys, we're gonna have to keep the chatter down for the video, sorry. Yep.
Is there any leniency for projects that the actual experiment takes multiple days or weeks to complete?
Yes and no. Yes and no. So I would say on a case-by-case basis, if you are communicating with me along the way, I've approved a project that I realize may have some issues, then absolutely there will be leniency in there. If I don't approve your project until January, whatever, because you email me over the break and you go, oh, this is the only thing I can do, and then you don't end up doing it, not much, no. Okay? Yes?
Are we gonna get rubrics for all of these?
Yes.
Because I was like, I cannot remember all of that.
Oh, no, I know.
Okay.
I know that. Yeah, I just don't have everything with me. All right, anything else? Okay, let's talk about the background research paper then. So this background research paper that's due Friday, December 13th. That is to be 5 paragraphs, okay, if they're big paragraphs. We're talking 2 to 3 typed pages of background information. And I would really like to know, because I found out first period that there were some people that have never written a background research paper before. And I assumed that that was something that everybody did in junior high. And so that really concerns me, and there were probably 4 people in that class, and I'd really like to take a poll. How many people in this classroom have never had to write a background research paper for any science project that they've ever done? And I'm not talking about the little 1-paragraph thing that I make you write for those formal lab reports. I mean an actual paper. Raise your hand if you've never done that. It's okay if you raise your hand, it's not like you're in trouble. No? Yes, kind of, you never have? I'm just curious, what school did you go to?
Kyrie.
A school in Kyrie District. Did they make you do science fair?
Well, for 6th grade. No.
For 6th grade, okay. All right, so that's good information for me, thank you. Because what I need to do is for students that don't have any background at all, I need to provide you with materials to kind of explain it more. And I need to know how many students I need to really account for with that. So the rest of you, yes, you will be getting rubrics. As far as the depth of instruction, that I'm assuming that with the exception of you, which we can always talk after school too, but I'm assuming that you guys all have a basic idea for how to do that, even if it's not super in-depth, and that provided with a rubric, it's not going to be a lot of hand-holding from my perspective with that. Now, I am going to be giving you Monday and Tuesday of next week, so we've got Thanksgiving coming up, so Monday and Tuesday of next week, I'll be able to provide you with in-class time to work on either test corrections or background research paper stuff, up to you, how you decide to utilize that time. Let's see, so Any questions about anything that I have written up here regarding the ISP? Okay, you know that you can always send me an email, you can always contact me if you need more help with anything. Are we good to move on? Okay, because today we get to talk about protein synthesis. And yes, I think protein synthesis is super exciting. It is one of my favorite aspects of all of molecular biology. You guys know I was a molecular biologist, right? So I love DNA. Well, DNA by itself, okay, that's just a molecule. And protein biochemists, I totally understand why they get really excited about their proteins. They get excited because their proteins do things, right? What kind of cool things do proteins do?
Give energy.
Okay, do they give energy? You're okay, I'm not really sure about how they would give energy. That's like the one thing I can't really think of a protein doing. Like out of everything, I'm like, yeah, I'm not really sure. But I'll say, yeah, probably. Because they kind of do pretty much everything, and I guess you could, oh, I know, you could break them apart during cellular respiration if you've got nothing else to break apart. Like you got no sugar, you break them apart. What's that? Okay, ooh, I like to use the word tertiary. So a tertiary source of energy. So like you run out of all sugars and all lipids, you can burn those proteins. Sure we can. What were you saying?
They're enzymes and they like do stuff themselves, they speed up reactions.
Very good. So we've got enzymes, super important with speeding up chemical reactions, because otherwise we wouldn't be able to regulate anything in a cell's metabolism. Yeah, Rishi?
They build things.
Oh, they do build things. Oh my gosh, they're so useful for building things. Generally as enzymes, but they can also make stuff up. Yeah, what else?
They've got muscles, hair.
Very nice. So there's certain structural component. So hair, nails. I'm not gonna beat my bones, but bones. So we've got a whole lot of different functions. What else? Oh, they're good for transportation. Oh, remember my favorite little walking protein? The little guy that holds on to that little vesicle and walk. What's he walking along? Proteins, right? The cytoskeleton, also made up of proteins. So we've got a whole lot of different jobs for proteins. So I do understand why protein biochemists get so excited about what their molecules do. Now, I'm a molecular biologist, so what does DNA, we study DNA, in case you didn't know that, by the way. What does DNA do that's so exciting?
Nothing.
Protein synthesis. It doesn't do anything. It just sits there, right? It's a double helix that just sits there. Like, that's not very exciting. I mean, come on, DNA, like, get with the program. But we can unwind it, right? And we can make copies of it. That's exciting. Not really. So what is the job of DNA? It's not to do all the cool, exciting things like proteins do. Even RNA gets to do cool stuff, not DNA. Yeah. Okay, think about for just a second how boring that is. Your whole job is to sit there and try not to change, because we don't want any mutations in you. So just stay the way you are. Occasionally open up so that we can make a copy of something, and then close back up and stay the way you are. Some more. So pretty boring idea as a job to just sit there containing information. So what does that mean then? It contains information. What does it really mean?
It genes your DNA.
No, I actually like where you're going. It genes your DNA up. Let's go with that. It genes your DNA up. What does that mean though? It genes your DNA up.
It like gives you all your things that you have.
Okay.
All your traits.
Traits. I totally know what you're talking about. So yeah, so genes are ultimately going to help code for traits. Now trait, that's like everything. What is that? To me, that's a really vague explanation too. Because what does that mean? It codes for traits? It means what? How is it doing that? It's a molecule that sits there. So how is it really coding for a trait? What's it actually coding for?
A protein.
It's coding for a protein, and we've already established that proteins do like everything. right? They are the doers. So they've got cool jobs, and the DNA contains the information to make them. So the only reason DNA is important is because proteins are important. And we need to make sure that we've got the right proteins for all of the thousands upon tens of thousands of jobs that need to be done inside of a cell every single millisecond in order for that cell to survive. So, yeah, the DNA is important because proteins are important. The DNA doesn't actually do anything except sit there. Yeah.
So like in a cell, like how fast do things go? Are they like in the videos? How like
So fast.
Like
It is so fast. Remember that one boring video on DNA replication where like, and now the enzyme is almost reached. No, it's like super fast. Like, I mean, you've got hundreds of thousands of things that are happening literally every single second. So, but you have to realize that atoms are small, really small, much smaller than anything that we can really imagine. And so it's all happening at such a sped up, teeny tiny rate that it makes up bigger stuff. And it's interesting because bigger things move typically more slowly than little things, right? And it's no exception when you look at atoms and molecules and the speed that they are able to go. But didn't I show you guys the DNA replication in real time?
Yes.
Didn't I show you that? And how fast that was going? Do you remember how fast that little ticker tape was just zipping out of the one side? It was super fast. It was very, very fast. So we have to slow things down for our brains to be able to understand what's going on in this respect. So back to this idea, we've got DNA that sits there, but it contains the information to make proteins that are super duper important. Now, does DNA actually make the proteins? So what is responsible in a cell for actually making proteins? What is it?
Ribosomes.
Very good. So ribosomes are responsible for making proteins. Where are ribosomes made?
protein.
I love the ribosomes. I know, right? I know. It's like this weird circular kind of thing. Okay, I want to know, so everyone think about it. So we're going back to like cells and cell parts and what makes what. Where are ribosomes made? Where are they made? Nope, they are not made in the cytoplasm. Where are they made? They are not made in the rough yard. Nope, your hand went down. They are made. Yeah, they're made in the nucleolus. Right? No, you were right. The nucleolus is inside of the so the nucleolus is inside of the nucleus. We desperately need them stable. So why don't you check the notes room and see if it's over there. She's actually in Mr. Hitter's room, so she's right there. Okay, so we've got the ribosomes that are ultimately going to be made in the nucleolus. What are they made out of? Ribosomes. What are they made out of?
Protein and DNA.
No, not DNA. Remember DNA?
RNA.
That's it. So we've got RNA and proteins that are going to be making up ribosomes. Okay, it's the ribosome, it's the RNA part of the ribosome that's so very important. And we call the RNA that makes up ribosomes, guess what? Ribosomal RNA or rRNA. Yeah, it's like, woohoo, okay, thank you for being Captain Obvious in that one. Yeah, so we've got rRNA, which is responsible for making ribosomes. Ribosomes are responsible for making what? Proteins. Okay, so we're getting this all tied together, right? So, let's see, so we got, how do ribosomes make proteins exactly? Oh, that's nice to have a word for it.
Protein synthesis.
Protein synthesis. Here, try and find the people that do that. Might try looking in the back, but I'm not seeing it. So protein synthesis. So, if we're thinking about protein synthesis, we know that ribosomes are necessary. We know that ribosomes are made up of rRNA and some proteins. And we also know that DNA contains genes. And genes code for proteins. So now what? Are we going to have ribosomes come in and attach to the DNA? Does that have the potential for damaging our DNA? And we hate that. Yeah, the second one, right? Yeah. Don't do anything to the DNA that might damage the DNA. So you think it's a better idea to maybe make a copy of the DNA so it can just continue to sit there? Because that's really all it's good for, is sitting there protecting that information. Now it does have to open up, make copies. That's what we're going to do. Now when we make a copy of a gene, we refer to that copy as messenger RNA, and we do it with RNA, not DNA. Now this requires, now that we're talking about RNA some more, that we understand some of the differences with RNA and DNA. So I'm going to go ahead and pull up next slide. I need to unfreeze it so that we can look at some differences between DNA and RNA. Because we've been talking about DNA. Oh, actually, before we do this, I would like you to revisit the idea of replication. So I want you to find this worksheet, one that says breaking the code. And I want you to turn to the side that says replication on it. So we're going to remember, you just had a test on this last Friday. You just did test corrections Monday and yesterday. So for this breaking the code, what I want you to do, I want you to notice that you've got 3 DNA molecules that are listed right here, here, and here. And I want you to write the complementary sequence in the lines provided. This is just showing me what you did on last Friday's test, just like that one short answer question, right? Showing me that complementary sequence. So go ahead and fill that out. What is the complementary base pair for T or thymine? Thymine is going to bond with adenine or A. And how about cytosine or C? It is going to base pair with guanine or G. So hold those papers up so I can take a look at them. Okay, never mind. Keep them on your keep them there, and I'll take a look. All right.
Wait, just the first one?
Okay. Okay. How's it looking? All right. Very nice. Keep going. All right. All right. All right. Looks good. All right. All right. Looking good. There we go. I need them all 3. All 3? All 3, yes. All right. Okay, so that's just our complementary DNA, strand to DNA. Awesome. Very good. So we remember those base pairing rules. So now let's take a look and see the differences between DNA and RNA. Here's something, actually, before we look at the differences, that's actually a pretty important idea in biology, this whole idea of the central dogma. That's the idea that DNA is basically a copy is made into RNA, and that copy and other RNAs are involved in then the making of proteins. So this idea that DNA to RNA to protein. Now, RNA is going to be necessary to actually replicate DNA. Do you guys remember why?
Because RNA primers.
Because of the RNA primers. So you can't actually make DNA without having some RNA. You can't make DNA without DNA. So this little arrow is indicating that DNA is necessary to make DNA, but you've got RNA that's going to be necessary for DNA too. Now that's something that we understood later. The original central dogma was simply a directional arrow that went this way. It went DNA to RNA to protein. And then once we started realizing that, hey, RNA is more important in like the just overall upkeep of DNA than we realized. So we've had to add some additional arrows in those directions. Yeah?
Where did the first DNA thing come from? Like if it needs
It's a
DNA
really
to make-
good question that we honestly don't have time to discuss. We actually think the original molecule was RNA, not DNA, because you can't make RNA with just DNA, and you can't make DNA with just DNA. So you have to have RNA for everything. We know that you can make DNA copies from RNA because reverse transcriptase that's included in retroviruses does it all the time. So we know that you can go that direction. But we have something called an RNA world hypothesis, which is what we think like the original molecule. So the original molecule for nucleic acids would have been RNA, and that would then explain why you can't make DNA except with RNA present. It's really interesting. We get into it more in AP Bio. So anyway, so I did have an interesting conversation though with a student yesterday, and it made me realize that we call it central dogma, but I don't think a lot of you necessarily know what the word dogma means, and that's going to be a problem in understanding what this is trying to illustrate. Can anyone tell me, does anyone in this class know what the word dogma means? In what context do you usually hear the term dogma being used? Is it usually something that you hear in reference to biology or chemistry or science in general?
I think history.
Think of history, yeah, or sometimes the term dogmatic. You could describe people as being, they're very dogmatic people. Does it mean they like dogs? No. You can also hear about dogmas in reference to religion too. So that's generally what I've heard most often, the word dogma in relation to. What do you think that would mean if a religion has a particular dogma? Yeah, that's it, exactly. So basically, it's the belief system or the principles that are followed by a particular organization, or in this case, division of science, i.e. biology. So you can almost think of it in some ways as rules, but that's kind of a really loose description. But when you talk about people being very dogmatic, then what does that mean?
They're like very strict on the rules.
Yeah, they'd be very strict rule followers, right?
Follow a set of moral principles.
Sure, and in those cases, they might consider them to be moral principles. So rules doesn't necessarily, when we're talking about dogma, it doesn't necessarily mean laws that are being followed. It doesn't necessarily mean law abiding. It could be rules or ideas or principles that are associated with a certain belief system. Well, in biology, we called it the central dogma because that was really how we thought everything was arranged. Originally, we said, wow, this is cool, DNA to RNA to proteins. Because there was debate even just 100 years ago as to, was it DNA or proteins that were responsible for heredity? And all the protein biochemists were saying, it's proteins, because proteins do everything. And there are some people that are like, why would you need DNA then? What does it do? What is the point of having DNA? If proteins literally do everything, including keeping the hereditary material and information, then what does DNA do? And so after multiple experiments, it was determined that in fact it's DNA that's responsible for the heredity aspect of life forms on this planet. Now notice I said life forms, because viruses can contain RNA, but they're not alive. So anyway, we have had to adjust and make some arrows that are kind of a little bit different from that progression from DNA to RNA to proteins, simply because as we discover more and more details about the actual procedures of how do you make DNA, for instance, then we're discovering more and more roles for things like RNA in there. So it's kind of interesting just to kind of look at this and how our ideas have changed over time. So based on what you see here, would you say that biology is a very dogmatic kind of discipline? Yes or no? Do you think that we strictly adhere to the rule of DNA, RNA, proteins in that one direction only? Or are we potentially open to change if there are additional information that we are able to gather? There is additional information. What do you think? Do you think biology is very dogmatic? Do we stick to our ideas no matter what?
No.
No. when you change it. And you shouldn't be dogmatic in science in general, because you have to always be open that the explanations that you have for something might not actually be the right explanations. I know, shocking. But you get new information, you're like, wow, that's weird. And then you have to think about it some more and maybe come up with a different explanation than what you had. All right. So genes. Segment of DNA that codes for a specific protein. And of course, enzymes are subclasses of proteins. Beetle and Tatum experiment showed that one gene codes for one protein. We've actually had to modify even that idea. So it used to be one gene, one trait. We realized that was totally wrong. Why do you think that's wrong, by the way? One gene, one trait. Do you think? What do you think?
Oh, was that chart right there?
Because of the chart right there, the
Each
Human
thing
Genome
on the
Landmark
device.
chart,
Yeah.
because there's a lot of genes on there.
Yeah.
Yeah? Okay. What do you think?
Because like there's two parents and
Oh, okay, so we're still talking about traits, we're not getting into genetics yet though. So hold that thought till we do. So I was asking this idea, why do you think one gene for one trait isn't appropriate?
I was wondering if the gene like sequences might overlap.
Could be. What else?
The DNA sequences are really, really long, so they can code for multiple.
But we could still say that you could have a really long sequence. I mean, if we were trying to say that you could have one gene, one trait, what's a trait? If we're gonna throw a word around, we probably should know what it means, right? What is a trait? Would be an example of a trait in a human.
Hair color.
Okay, hair color. Let's go with hair color. Eye color, sure.
Having a beak.
We don't have beaks, but I did ask for human examples, but, okay, sure. But sure, let's see, we're talking about organisms in general, not just humans, having a beak, although I'm sure if a human had a beak, that would be a trait that we could discuss. Yeah. All right, yeah, that's getting like super specific, right? But if we just went with something like more simple, like eye color or hair color, the examples that were first mentioned, do you think that's responsible, the responsibility of one single protein? Or do you think there might be more than one protein that actually factors into a lot of our traits? Think more? Because if you think about proteins that are responsible for catalyzing enzymatic reactions, for instance, or if they are even structural in nature, so if we go with hair color, do you think it's just one single kind of protein that makes our hair? No, it's multiple. And that's why typically for hair color, unless you're looking at, I mean, there might be certain kinds of exceptions. But generally, you don't discuss hair color in genetics classes as far as like dominant and recessive, do you? Because there are multiple proteins that are going to go ahead and make up the majority of our traits. So if we were to talk about one gene, one trait, that's just absolutely not true. One gene, one protein, it makes more sense. We've actually found out that when you think about quaternary structure of proteins, which we will be talking about again, that means you've got multiple polypeptides that are interacting. So technically, it's one gene, one polypeptide even. And you can mix and match different polypeptides to construct different kinds of proteins. It's pretty interesting. All right. Polypeptide is a chain of covalently bonded amino acids. Covalent bonds that are attaching amino acids are called peptide bonds. Now, do you think there's anything different between a peptide bond and a covalent bond?
No.
No, there's no difference at all. We just have this specialized term to describe a covalent bond that is linking two amino acids or more. I mean, obviously, we can link a whole bunch of them. But each time you're linking them together, we call it a peptide bond. Okay, we already talked about different examples of protein functions. We've got enzymes, transport proteins, signaling proteins, structural proteins, and a lot more. Oh, this is always fun. So finally, getting to some of the differences and similarities between DNA and RNA. And for this one, I like to be able to write on the board. All right, so we got our DNA and we got our RNA. Let's take a look at some of these. And yeah, I'm going to be starting to pick some sticks. So Might want to make sure that your textbooks are open so that you are able to find information if you are not already familiar with every single one of these things. Let's see, DNA I'm going to make blue, RNA I'm going to make red. Okay, Zashing, can you tell me subunits for DNA and RNA? What are they? I am so sorry, amino acids do not make up the subunits from for RNA or DNA. Can you tell me what does? Nucleotides. Very good. So the subunit is going to be a nucleotide. Okay, I can't write right there. No, that's supposed to be together, but I got a little silver thing right there. So a nucleotide here and a nucleotide here. So would that be a similarity or a difference? All right. Oh, I wrote the wrong color. Red RNA stuff. Okay, how about the sugar then? What sugar does DNA use and what sugar does RNA use? Dia.
Oh,
What sugar does DNA use?
deoxyribose.
Okay. And then how about RNA? And RNA is going to use ribose. What do you think the big difference is between deoxyribose and ribose? Jaden C.
The bases.
No, we're talking about sugars right now. So we're not talking about the bases, we're talking about the sugars. So I just said, what do you think the big difference is if we look at the word deoxyribose versus ribose? What do you notice is different between those two words?
The oxygen.
Yeah, has something to do with oxygen. Now if we have this D in front of it, what does that mean? If we think about it, does it have more? So would deoxyribose have more oxygen or less oxygen than ribose?
Less.
Less, because this right here, D, means that we've removed one oxygen from ribose. That's the only difference. It is exactly the same chemical structure. That one thing different then allows us to call this sugar ribose, exact same conformation, 5 carbons, same 1', 2', 3', 4', 5', same exact thing as DNA. It's missing one oxygen here. And so we call this one deoxyribose. And we're able to see that one extra oxygen has some interesting properties, doesn't it though? Like going all the way back to water and trying to figure out watery kinds of things, right? We've got some pretty interesting, oh no, we're gonna have lunch, aren't we? What time's our lunch?
58.
What time is it?
We have one more.
I got like less than a minute, okay. Let's try and get through this then. Bases, what's, Rishi, bases for DNA and RNA?
Adenine, so
Okay.
thymine, cytosine.
All right, A, T, C, and G. How about for RNA?
A, T, C, and G.
Nope.
Oh wait.
A, C, G is right.
And U.
And U, yeah. And so when we're thinking about this U, we're going to have to look at that pretty closely because just like up here, this deoxyribose is missing an oxygen, the only difference between thymine and uracil is that uracil lacks a methyl group. That's a CH3. That's it. That's the only difference. So that means, in my mind, it would make more sense to call uracil dimethylthymine. If we're going to say deoxyribose, that's dimethylthymine in my book. But that's not what it's actually called. It was a totally separate name for it, so we call it uracil instead. But I'll show you. The base pairing rules are the same then. That means thymine binds with what?
Adenine.
Thank you. Thymine binds with adenine, and then, so what is uracil binding with?
Adenine.
Adenine, right? Not thymine. So it's going to bind with adenine. How many strands does DNA versus RNA have? Carter, what? You have to pick this up after lunch. This is starting.
That's
Okey-dokey.
bad.
Okay.
This is like a pig.
Wow.
This is-
Maybe you can tell.
Okay, guys, the recording has already started for class. Tell me the class has started. So, please remember that you need to keep any kind of like side talk in the conversation to a minimum because it's going to pick up your conversations more easily than even what I'm saying because you are all closer to that camera than I am. So, unless you want every private conversation that you're having with your neighbor for everybody to hear, then I'd recommend not talking during filming. Okay, so we were in the middle of talking about similarities and differences with DNA and RNA. We had talked about both of them being nucleotides. We talked about deoxyribose versus ribose, and we talked about ATCG and how U is just like thymine, except it's lacking a methyl group. It's lacking a methyl group, so it's essentially the same kind of base. It involves the exact same base pairing with which other nucleotide? Adenine, yeah. So U is going to bind with A, just like T would bind with A. Biggest misconception students have is trying to make U bind with T. Does not happen. It's essentially trying to make two of the same kinds of hydrogen bonds take place in the camp. Yeah.
So it's missing a, I didn't catch what it was.
Methyl. So it's just a CH3 group. So it's a carbon with 3 hydrogens associated with it. Okay, so let's go back. We had started to talk about the number of strands for DNA and RNA. So let's go back and look at that. Daniel. All right. Did I already call on someone? We did answer it. I'm so sorry. Carter, then what was it?
2 and 1.
2 and 1. So 1 strand for RNA and 2 strands for DNA. Now the fact that RNA has just 1 strand, does that impact its ability to hydrogen bond with other nucleotides? Does it impact it, or can it still form base pairs? It can form base pairs. As a matter of fact, it does when we look at the structure of ribosomes being made up of what kind of RNA? Yeah, it's like in the name, right? And also tRNAs. We haven't had a chance to really talk about tRNAs that much, but what does the T stand for? Transfer, transport, translate. What are they transporting?
The amino acids.
The amino acids, very good. So with tRNAs, it is a single strand, and that single strand actually can fold back on itself, and this happens in the ribosome too. It folds back on itself, and so it's still going to follow that same kind of anti-parallel rule for base pairing, like what we had with the DNA, so I changed with Jeannie, right? So we still have that same kind of anti-parallel arrangement. So it just means like if this were all one long piece from here all the way over to here, only I couldn't have that, right? Because that would be 5 prime, this would be 3 prime, all right? So in that case, we would be able to fold over like this and in other different arrangements. And so that's how we're able to get 3-dimensional structures with RNA. DNA only has one kind of 3-dimensional structure. What is it? I heard it somewhere. Double helix, right? Double helix. I heard someone say it. Good job. So we've got a double helix with DNA, but we've got a variety of different 3-dimensional structures that a single-stranded RNA can take. It's just going to depend on how it's going to base pair with itself. So pretty interesting. All right, now let's look at the function of DNA. We actually already talked about this. So let's see, Daniel, back to you. What is the function of DNA?
To contain genes that code for proteins.
All right, so they are going to contain the genes. And so we can really think about the idea that they're containing those genes as being heredity, right? So DNA is going to be responsible for maintaining those genes, and so we're going to say DNA is responsible for heredity. We've already talked about RNA, right? At least when we're looking at RNA being a copy of a gene, right? So we can have a copy of a gene in messenger RNA. We can have ribosomal RNA, and we can have these tRNAs. All of these are going to be necessary for its function, which is, Gabe, what?
Protein synthesis.
Yeah. Making proteins. Protein synthesis. Anytime we make things in biology, we call it synthesis. We are synthesizing something. Yes?
So like, are these proteins like the same proteins that you get from like a protein bar?
Absolutely. Because remember, all of your food is made up of organisms. So if you're eating a protein bar, you're eating up, proteins that were made by the organisms that were basically crushed up and put into those protein bars. Yeah. You can also isolate proteins from organisms, and then you can say, hey, this is going to be like purified protein, and you can make it into a powder, and then you can add purified protein to things. So this is done with soy a lot of times. So there's like TVP, textured vegetable or texturized vegetable protein, where it's actually protein that's been isolated from soy, and then it's powdered, and then you can add it to different things. You can do that with peanut flour. You can do that with a lot of different things that contain protein. So yes, all of your proteins. If you hear the word protein, it came from a life form. That is the only thing that makes proteins. You don't have random proteins being generated out in nature. Organisms make that. That's why we learned about the macromolecules in the first place. Because all of the macromolecules that we talk about are created by organisms. The entire idea of organic chemicals, those organic chemicals ultimately have derived from life forms. Now over time, if you crush them up and stick them underground for a long time, you end up with petroleum rather than the kinds of organic molecules that are actually going to make life forms able to function. By the way, even though petroleum is organic, should you eat it?
Yeah.
No, you should not eat petroleum. I'm talking about gasoline that goes in your car. No, you don't eat petrol.
That was one time, okay?
No! No! Never, ever drink gasoline. How about that? Your words of wisdom for the day. Yeah, no, it's a terrible idea. So then it is an interesting question. So if you can't, just drink or eat all organic things, what value does it have putting organic as a label on something? Because, by the way,
Makes the
drink.
price go up.
It does. Yeah, organic molecules by themselves are not good for you. Organic molecules simply mean that they were derived from a life form. There are plenty of organic molecules that are poisonous and even deadly. By the way, if you drink gasoline, that is potentially fatal. So shouldn't do it, ever. Not even little bits of it. Not even like, I just had this. No, you don't drink gasoline, okay? So we'll have to have, when we talk about GMOs, when we talk about some other kinds of interesting things later on, we'll kind of attack this idea a little bit. Because the organic label that you get in a food store has absolutely nothing to do with chemistry. Nothing at all to do with chemistry. So the organic term in chemistry that we have been using from the beginning, from the beginning has been in the chemistry definition form. That means it's derived from life forms. Okay, let's see, process formed in. That's a little bit confusing. So basically it means, how are we forming DNA? How are we forming RNA? So what is that process called? Naharka, what is it called when we make DNA? Yeah, we call it DNA synthesis, or we call it DNA replication, right? We call it DNA replication. We call everything, anytime we're making anything, it's synthesis, right? So DNA replication. This is terrible having this metal bar right in the middle of the word. So we call it DNA replication. What do we call it, Rena, when we are making RNA? Using DNA as our template. What is that called? You got your book open, right? You could even look on this worksheet right here. See, we have DNA replication right on there. And then we have something down here which involves the making of RNA. What's it called? It is called transcription. Transcription is the process of making RNA from a DNA template. So we call it DNA replication when we're replicating DNA, and we call it transcription when we're making an RNA copy from a DNA template. And you kind of think of it as transcribing. What does it mean? What did scribes do?
Write down what is said.
They write down what's said, right? So like when you think about really old, like medieval time, the scribes, what were they doing? they were copying, right? They were copying. They would have a book, and they would copy down what was in that original book. That was back in book burning days when you were allowed to have books. So they would transcribe what was actually said in the original book. They would make a copy of it. And that's really what we see with transcription, where we're getting RNA from a DNA. So you would have that original template, and then you would have a copy of it in RNA, which is slightly different, right? You can see it's slightly different from DNA. Okay, so location in eukaryotic cells. We are talking about eukaryotic cells. Nick, where is DNA found?
In the nucleus.
It is found in the nucleus. Very nice. Yeah, we don't have DNA just randomly roaming about. Why would that be bad if our DNA was just randomly roaming about?
A lot of processes going up the cell membrane.
It could get damaged, yeah. Oh, is that what you were trying to say? Thank you. So yeah, it could get damaged. But then that leads to an interesting question, which is, which we'll deal with in a second. Isabel, what about RNA? Where's RNA found?
We found like inside the like it can be found in the nucleus, and also in the cytoplasm.
Yeah, it can be, right? So we're going to find it in the nucleus and in the cytoplasm. We can say everywhere, but if I say everywhere, I don't mean like inside the Golgi, and I don't even mean inside of the endoplasmic reticulum, because when ribosomes are attached, they're attached to the outside. But so we'll say nucleus and cytoplasm. All right, so nucleus and cytoplasm. Okay, so please make sure that you've been taking notes on all of this. And I believe there's even a chart on the one worksheet packet that I gave you that looks somewhat like this. Yes, Gabe?
Can you also find it in ribosomes? Can you put that down? RNA?
Yes, because ribosomes are found in the cytoplasm, but absolutely you can write ribosomes down too, because that's what a ribosome is essentially made of. Wow, let me put this back down though. Okay, so when we are looking at ribose, okay, here with the ribose, and remember we've got carbons anytime we're drawing an organic molecule and we have like an angle, we have a carbon there. So that means we have a carbon here, 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 and here. So that would be the 1', 2', 3', 4', and 5' carbons, just like we had those 1, 2, 3, 4, and 5' carbons in deoxyribose. Now this is ribose. What does ribose have that deoxyribose does not have? It has this oxygen right there. Oh my gosh. As soon as that oxygen is gone, oh, I can't cover it up because it's light. As soon as that oxygen is gone, then that's deoxyribose. That's the only difference between ribose and deoxyribose. If we look at the bases, so adenine, guanine, cytosine are all going to be identical to the nucleotides that we have in DNA. Those are totally the same. As a matter of fact, that's the same adenine that we would have in ATP, so adenosine triphosphate. Has this exact adenine as well. By the way, ATP is ribose, not deoxyribose. Okay? So it is an RNA nucleotide. If we look at uracil, remember I told you there was one difference between uracil and thymine. What is the difference between uracil and thymine?
A methyl group.
Okay, there's a methyl group that's going to be different. Uracil, notice, does not have a CH3. Do I see a CH3? I do not see a CH3 here. Or what they might have shown it as is just a single line going up, because that would have been indicated that a carbon would be there. So in thymine, you would have a CH3 right here. That is the only difference between thymine and uracil. Notice that over here, these portions of that base are what's going to be involved with the hydrogen bonding to an adjacent nucleotide, so to make one of the rungs. And by the way, uracil, what's it going to base pair with? Adenine. Now, it'd be kind of cool if we had them oriented in such a way that we could show base pairing, but they are not oriented in that way, and that's not something that we have to be able to demonstrate, the actual hydrogen bonding. I'll make you do that in 85. So some things we can see. All right, so here are our bases for RNA. And then notice then we've been talking about the different parts of nucleotides. So what is this part right here?
That's a phosphate group. What is this part right here?
Sugar.
That's the sugar. Now, is the phosphate going to be different between DNA and RNA?
No.
No. But the sugar is going to be different, right? Because this O right here is going to be missing a deoxyribose. And then is this base going to be different or the same? It's going to be the same between DNA and RNA. This is going to be the same. What about this one?
Same.
Same. This one? This one? Oh my gosh, because why? There's a methyl group right here in thymine. Oh my gosh, it's so different. All right, so there are our nucleotides. Is a nucleotide and a base the same exact thing? No. So if we go back, here are our bases. Here is our nucleotide. A base is one part of a nucleotide, right? Okay, so here are the three main types of RNA. We've got messenger RNA. That's where we have, so taking the genetic message, we're going to make a copy of the gene into RNA. That's going to be called the messenger RNA. That's going to have to go out. That's going to contain the sequence necessary to actually build a protein. We've got ribosomal RNA, or rRNA. That's going to be making up ribosomes. Where is that made, by the way? Ribosomal RNA, where is it made? It's made in the nucleolus. It's made in the nucleolus. Let's see, transfer RNA is going to transfer the appropriate amino acid to ribosomes when instructed. What's going to be responsible for the instruction? What's responsible for actually instructing a tRNA when to come in and go and, give an amino acid? What's in charge of that? No, think about it. Whose job is it to make a protein?
Ribosome.
Ribosomes. Ribosomes are responsible for making proteins, right? So the ribosome is going to ultimately instruct the tRNA when to come and give its amino acid. We're going to act it out, by the way, on Friday. So you guys are going to each, well, not everybody, so half you. So some of you are going to be tRNAs, and one person gets to be the ribosome, and you're going to actually have to carry your little amino acid if you're a tRNA, and you're going to have to go where the ribosome tells you to go. It's pretty cool. Okay, so this is kind of looking at what does it look like. And don't forget, this is going to be on Infinite Campus. I'm going to put that on Infinite Campus by the end of the day. So here's our mRNA strand. That's going to be the sequence of the gene that's just been the complementary base pairs of the template DNA. Here's our ribosome made up of rRNA. And yes, it does have some structural proteins, but it's the rRNA that gives it the ribosomal characteristics that it needs to actually be able to direct protein synthesis. And then here's our tRNA. Notice one strand, but it folds back on itself. In every place you see it kind of folded like that, that's where hydrogen bonding is occurring within that strand with other parts of itself. And then you get an amino acid that's going to be attached up at the top. It's pretty cool. Okay, so right now with your partner, everyone needs to have a partner. If you don't, because you are a single person sitting at a table, I think, Daniel, that's just you, then I need you to talk to the people that are in front of you. Think, pair, share. What is the difference between genes and DNA? What are differences between RNA and DNA? Now, I think you have those written down, but talk to your partner right now about those differences. 60 seconds. Hey, basically, you asked me. Yeah, exactly. Keep it it's like scissors. It has a use. It doesn't do anything by itself. Okay. So, Una, what is the difference between genes and DNA?
Genes are like a little part of DNA, or like a few like
nucleotides, whereas DNA is like the whole thing.
Very good. So DNA is going to be the molecule ultimately that makes up genes, but genes are just a specific little portion of that DNA that's going to code for a particular polypeptide or protein. Okay, now what are differences between RNA and DNA? I'm going to call in several people for this. Erica, tell me one difference.
DNA is double-stranded and RNA is single-stranded.
Very nice. So we got double-stranded DNA, single-stranded RNA. Oh, like D, double DNA, D. Yeah, double D. Double toilet trouble. Patrick, what is another difference between RNA and DNA?
DNA is deoxyribose and RNA is ribose, so deoxyribose is missing one oxygen.
Very good. And so we're talking about the sugar that's making up the actual nucleotides. Has a difference. Robbie, what is another difference between DNA and RNA?
DNA is 2 strands.
No, we mentioned the 2 strands, 1 strand, and we mentioned deoxyribose versus ribose.
So the process of DNA is DNA replication.
Okay.
RNA is transcription.
Very nice. So we have 2 different names for, even though they're both using like a template strand of DNA to be made, we call it DNA replication when DNA is being replicated and we're using both strands. For RNA transcription then, we're only going to be making a copy of one of those DNA strands, not both. So we only get one of those strands and it is called transcription. We'll look at that in a little bit more detail today and a lot more detail later. Someone had a hand up over here. Was it you?
Oh,
No.
it's just changed to the motion.
Oh, okay, I'm sorry. Let's see. There was one more difference between RNA and DNA. Nana, do you remember what it is?
Yeah, for the basis, RNA.
Very nice. So we had uracil in RNA and thymine in DNA. And is there a huge difference between uracil and thymine?
No.
Yeah, there's a mountain of difference. It's like so. I really wish we could just call it dimethylthymine. That would just can't do it.
Is uracil a purine or a pyrimidine?
Uracil is going to be a pyrimidine, just like thymine is. Okay. So, and the way to tell, it's a good question. The way to tell, anytime you're looking at nucleotides, let's look at specifically the bases. If you notice, adenine and guanine both have 2 rings. See the ring structure here and the ring structure here, how they're slightly bigger compared to uracil and cytosine, and of course thymine is the same thing as uracil, right? So we call these 2 ring structures purines, and we call the single ring structures of uracil and thymine and cytosine, we call those pyrimidines. So good question. Okay, so we're going to look at transcription, but we're going to look at it kind of quickly today. So I'm going to kind of breeze through a couple of these slides. We'll go back in detail later. I want us to get a nice overview today. So first off, we're talking about transcription. What is it? We're talking about building RNA using DNA as a template. Okay? You excited about that? All right, where does it take place? Well, I mean, where is it going to take place? Where are you going to make a copy of a DNA? It's going to have to be, right. In eukaryotes, it's going to be in the nucleus. Now, it's going to be in the cytoplasm for prokaryotes. Jewel, can you tell me why? Good answer. Good. Talk about getting put on the spot, huh? Yeah, because prokaryotes do not have a nucleus to do that. Now, it's kind of interesting because that means their DNA is just sitting there, in with everything else, like a big giant soupy cell, where you don't have all those nice little compartments like we have in the eukaryotes. But it's also a very, very tiny cell, so there's just not as much stuff there. Still has all the necessary chemicals for life, still has the potential problem with DNA. Now their DNA is also slightly different. Do you remember from your test that you took on Friday? What is different between prokaryotic DNA and eukaryotic DNA? Carter, do you remember?
Prokaryotic DNA is like circular.
It is circular. And the fact that it's circular kind of protects it a little bit more than having linear DNA. Because with linear DNA, those ends are just kind of flopping around. And we happen to have something called telomeres, which kind of protects our linear DNA. They don't need to worry about that. So we look at transcription kind of briefly then. Here's our DNA. Now some things are going to have to be similar with transcription compared with DNA replication. We're still going to have to open up, helicase is going to have to come along, it's going to have to open up that DNA. It's going to have to unwind it. It's going to have to unwind the two strands. Now there's a big difference now. Do you remember in DNA replication, we had replication happening on just one strand? Did replication happen on just one strand? DNA replication? No. Remember we had leading and lagging strands for DNA replication. And so both strands had to get replicated. Both had to act as templates, and then both ended up going into the new DNA molecules that were being made, right? So you ended up with one old and one new strand in each of the new molecules. Now transcription is different. We open up the DNA, and only one of those DNA strands is going to be used. Depending on the gene, it might be one side or the other side. It doesn't mean that we have got one strand of DNA that always is the template and the other strand is just always there. It can be on either strand, but it's never on both. Yep?
What's the difference between the antisense strand and the sense strand?
The antisense strand is the template strand, okay? And then what that means when you see the fact that it says sense strand, if you notice the sense strand has the same exact sequence on it as the RNA transcript will. Now this RNA transcript here, we're going to call this messenger RNA. Now all transcription, whether it's making ribosomes, ribosomal RNA, or tRNAs, it's going to happen in the same way. But in this case, we're making this is a messenger RNA. And so it's called sense strand, not because it's used, but because you can look at it and go, oh, if I just replace every T there with a U, I now know what the sense is of my gene. So you can look at this and basically it's going to tell you what your mRNA is going to look like. It's a little bit more complicated than that because, and we'll deal with RNA processing, messenger RNA processing at a later time too. In prokaryotes, it works exactly like this. In eukaryotes, you are going to have to cut out some of these pieces in the RNA transcript, so it won't end up looking identical. But that's why we have antisense and sense written there. So the antisense strand is the template strand. But again, these are going into little nitpicky details that we'll deal more with later. I want to see a big overview picture today. So there's our RNA transcript. This globular cluster here is RNA polymerase. We will talk more about RNA polymerase later too. Okay, that's kind of getting into those nitty-gritty details. Because like, where does RNA polymerase bind, and how does it start? And you know what's cool? You know what RNA polymerase can do that DNA polymerase can't? Start it. Can DNA polymerase start replication?
No.
No, it needs help. It has to have RNA primase come on and put some primers on because it can't start it. Aw, poor DNA polymerase. Look what RNA polymerase can do. So let's go on and look at one more thing. Oh no, we're gonna skip over the next couple of slides. The next couple of slides are basically talking about starting, making it longer, and then stopping. But what I want you to do right now is on this worksheet, the same one that you wrote DNA replication, so you wrote the complementary strands of DNA, I want you to now transcribe. You're going to be transcribing the template strand of DNA that is provided for you into an RNA transcript. So what do you have to be careful of when you're writing this, Rishi?
When you're writing, you should not.
Right. So right here on the template strand of the DNA, so the antisense strand, and that template strand, when it says A, you're going to write U. Now when it says T, do you write U? No, you write A. All right. G, then you're going to write C. Okay? So go ahead, fill those three in. I want to see that you're doing this correctly, okay? Now, one thing I'd like you to do with this part is I'd like you to, it says leave a space, but you guys have already written it. So I'd like you to draw an up and down, so a vertical line between each 3 nucleotides. So you're making mRNA right now, and after every third one, I want you to draw a little vertical line so you're separating them. Good. Is that a G or a C? Because this looks like a, okay. Ooh, very nice spaces on that too. That's what I directions. Good. Okay, so if we are, and go ahead and continue with that, I'm going to skip through the next couple of slides. So initiation is just a fancy word for starting. Elongation, fancy word for making it longer. Termination, fancy word for stopping, right? I've always wondered, why don't we just call it start, make it longer, and stop? I don't know. We're biology. We have to be fancier than that. So initiation, elongation, termination, it is. We're not going to be 140 right now. We're not going to be doing those things. We're going to do something else we're working on. All right. So let's go ahead then and think about this. While you guys are finishing up that mRNA. So great. Now we have a copy of our gene. It's in RNA. Wonderful. We've got our ribosomal RNA making up ribosomes. We know ribosomes are going to be making those proteins. We've got tRNAs that are going to be attached to amino acids, and they're going to be able to bring them over to the ribosome, and they're going to stick those amino acids where the ribosome tells them, and the ribosome is going to be catalyzing peptide bonds between those amino acids. And how does any of that work? And here's another thought. When we think about any of this, it's kind of weird, really. Because we're talking about trying to make a completely different kind of chemical sequence from a completely different kind of chemical sequence. Are nucleotides and amino acids the same exact kind of molecules? Are they the same? Do you have a phosphate group on an amino acid?
No.
No. Do you have a sugar in an amino acid? No. And that's where a nucleotide is really kind of weird. Nucleic acids are odd because they have another macromolecular group in them. So in order to have a nucleotide, you have to have a carbohydrate present that you can make a nucleotide out of, right? That's kind of unusual. That's kind of interesting that it has to be done that way. I have a question for you. How many of you speak another foreign language? Like either at home or with extended family, or you've ever, you've got another language that you periodically have to use. Okay? Now, it's kind of interesting when you're speaking in a different language. And is it easy, would you say, to kind of like slip back and forth? Like, there's some people that you have to speak English with, and then some people that you have to speak this other language with. And you can hey, this is my friend, I have to talk to them in English, and then this is my dad, I gotta talk to him in whatever language it is you have to talk to your dad in. And so you have to kind of go back and forth. It's kind of interesting in your brain, because it requires a certain fluidity of thought. That is one reason why colleges like you to have a sequence of foreign language instruction before you go to college, because it is very good to be able to kind of slide through different languages that way. And I'm assuming everyone is taking it. Okay, raise your hand if you've ever taken a language or are currently taking a foreign language. Okay, so I thought that would be everybody. All right. Because what's interesting then, when you're taking a foreign language, do you automatically, when you first start learning a new language, understand everything that's someone who's speaking the language. No, what do you need to have in order to be able to understand someone who's speaking a different language to you? What do you have to have? You're going to have to know what the words mean, right? Wenn ich auf Deutsch reden, können Sie mich verstehen? Ja oder nein? Ist es einfach oder schwer? So, if I speak to you in German, if I try to teach this entire lesson to you in German, are you going to be able to understand me?
Yeah. I speak.
So I literally just said, if I speak to you in German, can you understand me?
No.
And then I said, you know, would you say it's easy or difficult? And you said yes. Somebody said yes. It is. Whatever you said, yes. It's whatever. So you have to have vocabulary, right? You have to know the rules that are involved. You have to know how to interpret what's being said into a language or a thought or a concept that makes sense to you. Well, guess what? When we talk about going from the language of nucleic acids into the language of proteins, we have to know the rules. We have to know the vocabulary. How do you do it? How does that work? And if you're off and you don't know them exactly, then you're not going to be able to understand what's happening. So in this case, we then talk about going from nucleic acids in the form of RNA into proteins translation. It's how do you make a protein from nothing but nucleic acids. And the cool thing is, we got the code. We cracked the code. It's amazing. This is just looking at translation. So here we look at an mRNA molecule. So just like the ones you wrote down on that worksheet just now, if we've got an mRNA molecule like that, then notice we have this term right here called a codon. And we're going to look at this really carefully. Now I made you, on your mRNAs, I made you separate each three nucleotides with a vertical line, or space if you already put a space on there, right? And I did that because the messenger RNA is broken into triplets. Every 3 nucleotides means a different amino acid. So every 3 nucleotides means a different acid. And so in the mRNA, we call this code a codon. So you get it, codon, or code. All right, that's our codon. Now, this is very, very important. I need you to write it down somewhere, even multiple places. Codons are found in mRNA. Now, if we're talking about, Rowdy, I need you to write that down. Codons are found where? mRNA. This is important because we're talking about different kinds of nucleotides. I mean, we're talking about DNA, which strand, the template, the non-template, sense, antisense. Which one? Are we talking about DNA? Is a codon found in DNA? No, codons are found where? Right. So we're not talking about the tRNA. We are not talking about the rRNA, and we are not talking about either strand of the DNA. A codon is found in the mRNA. It's important that we are precise in our language, because otherwise it gets really confusing. Because what strand of nucleic acid would we be talking about? In this case, a codon is always referring to mRNA. Good job. So let's see what the code is then. Oh, we're talking about editing RNA later. Okay, but this is cool. I like this. So basically what that means is if we've got an mRNA, the ribosome is going to come. What's the ribosome do? Makes proteins, right. So ribosome is going to come and attach to the mRNA. It's going to direct the synthesis of proteins, because that's its job. It's going to make proteins, right? Well, so the mRNA is going to provide the sequence. With the codons, right, that's going to say which amino acid needs to get in each and every position. Here's your code. All right, so here's how to read mRNA. So the codon has 3 nucleotides in it. And take a look, you've got this chart. You got this chart right here, okay? So when we look at it, notice we've got a first letter here, second letter here, third letter here, right? So I want you to look at the mRNA strands that you just transcribed right here, okay? What did you have, Megan? What was the first codon that you had in the first mRNA that you have? AUG. So we would need to come to this is the genetic code, by the way. When people talk about the genetic code, this is what they're talking about. This is what we can look at to determine what a protein sequence is going to be with nothing more than knowing what the gene is. So we can look at this and go, oh, we can figure out the protein sequence from that. That's pretty cool. So here, first letter, A, right there. See it? Second letter, U. So we know that we're in this box somewhere. Third letter, G. Go all the way over. And notice here is the amino acid that will be coded for by AUG, methionine. Also notice it says start. Notice for me right now, all 3 of your mRNA molecules that you just wrote start with which codon? AUG, which is going to code for methionine. Now just because methionine starts an amino acid sequence does not mean that you can't use it later. So you can find AUGs anywhere in a mRNA transcript, but they do always start the entire thing. But it doesn't mean every time you have an AUG, oh, we gotta start again. It doesn't work that way, that would be horrible. Methionine is useful and we do like to use it in proteins, so it does get used. So we've got AUG right here. Now, how is methionine going to get to the mRNA? How is the ribosome going to attach that methionine from that tRNA to an adjacent amino acid from a different tRNA? What's going to happen? How does that work? What do you think, Dave?
So the codon attaches to the mRNA, or the anticodon, which is on the tRNA, attaches to the codon on the mRNA.
So base pairing, huh? Let's go back and look at this and see for a second. All right, so if we have AUG right here, and we know that that's coding for which amino acid? AUG? Methionine. So that means we need to get a tRNA there. A tRNA that can definitely recognize AUG. And that tRNA that recognizes AUG better have methionine on it, because that's what it needs anytime you've got AUG in the mRNA codon. So what we can see then is a tRNA. This is going to be a terrible tRNA, by the way. Ooh, it looks like an upside down clover. So on the tRNA then, if we've got our AUG on our mRNA, then our tRNA needs to have a U, an A, and a C. Okay? Now what that means, ooh, I'm gonna color code better. Here, I'm better at it. I'm gonna do this mRNA. So this is gonna be mRNA codon. Right? Because the codon is found in what kind of nucleic acid? Specifically mRNA. Very nice. And so the tRNA, the fact that it can base pair here, oh, by the way, this tRNA is going to have methionine as an amino acid attached to it physically. So this tRNA then is going to have an anticodon that is able to form base pairs. So it can form base pairs. What are base pairs formed with? What kind of bonding? Hydrogen bonding. Yep.
So would it also work if the anticodon is T instead of U?
It can't, because all of the RNAs are only going to have U's, and they are not going to have T's in them. So no, because it's all RNA based. Oh, I just drew this big giant brown thing. What do you think that's supposed to be? Yeah, we gotta have a ribosome, right? Ribosome. Which has what kind of RNA in it?
RNA.
Yeah, we got our rRNA, okay, and our ribosome. So basically then that means on our mRNA transcript, we're gonna have the ability for within the ribosome, and the ribosome has like these little compartments that the tRNA can just kind of come and fit in. And then if that tRNA has the appropriate anticodon, only if it has the appropriate anticodon, it can then base pair, okay? And then that means it's positioning its amino acid in a perfect spot for the ribosome to catalyze peptide bonds, so that covalent bond between adjacent amino acids. Now, again, we're going to act all of this out on Friday. So, AUG. What about GGC? What amino acid is that coding for? GGC. Hold on, let's see. So if we're trying to read it, GGC, we've got G here, we've got G over here, and C right there. So glycine. Glycine is what is being coded for with GGC. So we would have, so if GGC was the codon, for glycine, what would the anticodon of the tRNA have that's attached to glycine? So GGC, it would have CCG in the anticodon, right? Now here's what I want you to do. So flip to the back side of this paper, and I want you to notice you've got two different things that you need to do with the mRNA sequences that you wrote down at the bottom of this side. At the top, you're going to write the anticodons for that mRNA, and then underneath that you're going to have the translated, as we call this process translation, when we go from nucleic acid, so RNA, into protein, we call that translation. So I want you to write down what amino acids are being coded for by the mRNA sequences you have. Be careful when you're doing that to not use the anticodons that you're doing up here at the top of this page, okay? So the anticodons, how are you going to figure that out? How are you going to figure out the anticodons? Rishi?
The base pair of the.
Because you're looking at the complementary base paired sequence that would match up with these mRNA codons, okay? Do the same thing, by the way, with the tRNAs, the anticodons. Separate those so that you know that each one of those has its own separate tRNA. And that tRNA is going to be attached to what? tRNAs are attached to, what are they attached to? They have to be attached to amino acids. So go ahead and fill in what the anticodon would be. Are you going to have any T's at all in these sequences?
No.
No T's. And then you're going to translate it using your genetic code right here. Be careful not to try to translate your anticodons. Just translate your mRNA. Because codons are found where?
mRNA.
On mRNA. So make sure that you're doing that, okay? I'm going to give you a few minutes to go ahead and finish filling that in. Okay, well, let me see how you guys are doing. Go ahead and start writing stuff down.
I just like
You're the anticodon now. Okay, so see these codons that you wrote? You're going to write the complementary sequence. That's going to be the anticodons. That's the tRNAs. They're going to bind up with each of those, okay? And then you have to translate each of these here into the different amino acids.
So you're telling me you don't remember doing this? So once it stops, we just stop.
Yeah, you don't have, there's nothing else to add. Correct. Okay, do you see how to do it? anticodons are found in the mRNA. So see where you have AUG, that's what you use, AUG. So the first one's going to be methionine. Don't use this, use the other side. Let's go. Remember, don't use the anticodons. So what are you going to be doing to figure out the polypeptide? Yes. Nice, yeah, you got it right. Why'd you erase methionine? Well, methionine's right. So, yeah. So, yeah, so you're good. So that's methionine, and then you need GCC. Glycines. Methionine, glycines. We were doing it right. Look at you. Are you already finished with everything? Oh. Okay, but I need you to practice this one. You're to make sure that you can do it, okay? And then you're absolutely welcome to work on it. I need to make sure that you guys know what you're doing. No. Although it does work out that way, you're supposed to be complementary base pairing with this. So the tRNA is going to come and base pair with that because it's going to have UAC in that spot. Does that actually match up with this? It does. And that gets into the sense and anti-strength sense idea too. So when you're looking at the anti-sense, that's the complementary sequence that is the template strand that makes this, it's going to have the same sequence as the as the tRNA does, because both of them have to complementary base pair with that. And Julie, are you saying how to do this part too? That's okay. I just want to make sure that you're knowing how to do that. So you're going to be taking these sequences, so like AUG. Yeah. Okay, just want to make sure you're okay? Yeah. Why are you not doing it? Yeah, okay. So see where you have AUG here? Okay. So this is your mRNA. That's going to be your codon. So that's what you're going to look at when you're trying to make your polypeptide here. It's your AUG. Okay, and this is what I'm trying to do. This anticodon for the mRNA, that's going to be on what?
Yeah, we can see this here.
The anticodon is found on the
Of this and
Hey,
right.
remember, you don't use anticodon, right? So it is going to, so basically that means what tRNA is going to bind to AUG? tRNA anticodon is going to have to be UAC, right? That's what you write down right here, UAC. And so then you're just writing down UAC. How about here? Where's the anticodon is the tRNA that combined with that? Exactly. And here? Very good. So that's what you're writing on that part. No, that's fine. My son, two of my sons have top notes. Yeah, one, it'll be like 6. Yeah, or like 7. Or 6.
Oh, really?
We do, it's really interesting. I had no idea what was really involved with it. But yeah, it's not a normal kind of job. And they say that they get like thousands of them. And they look higher.
I know. I'm very angry. I am angered at this moment.
It's that with like the other.
Yeah, you went through all, yeah. It was yeah, he's sort of training back. Something like that, Neil just started like working, like not being trained. So you understand how to do this?
Yes.
Right, so you have to match, but you're not matching with this, right? You're matching over here with this. Okay.
Okay, so you
Bobby,
can.
how's it going? Okay, now I need you working not on this right now, I need you to work on this sheet. Okay, so this is, so here, all of this stuff that you did right here, you're going to actually translate this. You're gonna translate this here, okay? So you're gonna, so A, U, G, there should be A, G, right, that's fine, they write the fine. And then you look at the next one, GCC, so you go over here, so G, here's C, so GC, here's Make sure I did it right. Make sure it's right. Yeah, oh guys, I was saying glycine, it's not glycine, is it? I was looking at G, like over here. That's not C, that's G. So when I was saying glycine, it's not, it's alanine. My bad. Didn't you say it was alanine yet earlier when I was saying, oh, GCC, what is it? I'm like, G, C, but that's G, that's not. Make sure you're paying attention to those kind of things, that's important.
Appreciate it. Sorry for making fun of you when you got that wrong.
Okay, now if you finish all this, fantastic, because I would really like you to work on this other worksheet while everything is fresh in your head. Okay, well, all this information is totally fresh in your head. Once you've finished with the practice. Now, I would like us to, has everyone finished at least the first translation of polypeptide number 1?
Yes.
Yeah.
Doing them all like in a row, so.
So that makes it kind of hard, because you're doing them just straight up and down in a row like that. Okay. Well, I tell you what, let's just go ahead and I'll give you a couple more minutes to work on that so that we can at least go over the polypeptide number 1 and make sure that we're doing it correctly. And like I said, if you are finished, then please start filling in this worksheet. You do not have all of the information necessary to fill in this entire worksheet, right? But you can certainly start where you're looking at list and describe the function of the 3 types of RNA, filling in the chart for the number of strands, sugar and nitrogen bases for DNA and RNA, functional differences between RNA and DNA. Those are all questions that you can already start to answer. It's going to be a work in progress. Don't forget, you've got a vocab quiz on Friday. Next time I see you, you've got a vocab quiz and your vocab is due. Okay. That's easier for you. It seems like more work to me, but if you just don't like paper back and forth. So you got your MCAS, right, that you already figured out. You've got and then what you're doing right now is you're going through and assigning all of those, that's fine. That's
Oh, yeah. Just remember that right here, you're also gonna be listing out an anti-codon. So the tRNA that's attached to the kinase is gonna have to be able to recognize the AUG by itself having UAC in its anti-codon, right? Yeah, right, because it has to bond somehow, so those hydrogen bonds are gonna form because it has an anti-codon that's gonna match up with that codon. GCC would be what? CGG, yes, exactly. And so then that's what you write it down.
You guys are starting
Good, I don't know. You guys have any questions for me?
Scott, do you need recording anymore?